share our work with all of you. My lab works on neuromuscular genetics, and over the years, we have discovered a lot of disease-causing genes that result in neurological or neuromuscular disorders. And right now, our efforts are focused on developing therapies for many of these diseases. We use zebrafish and mouse model to model some of these disorders, and I'm going to talk to you why these are such a wonderful models to study disease and eventually developing therapies. So I'm not going to talk about a lot of background because Seema already gave a wonderful background on TANGO2 Deficiency Disorder. But just to remind everyone, this is a disease which is a recessive disorder. So you need 2 copies of the mutation in TANGO2 gene for the disease to appear in the patients. Disease onset can happen from the congenital to the adult stages. And there are a number of clinical phenotypes that are associated with the disorders, from delayed milestone cognitive disorders, rhabdomyolysis, and this is something I'm going to talk more about, which is the muscle breakdown and cardiac arrhythmia. So most of the genetic disorder, when that happen, you have the loss of function of a protein, and because of that, there can be a lot of changes happening in the body. But TANGO2 disorders is quite complicated because not only we have the absence of TANGO2 gene, these patients are extremely vulnerable to a lot of environmental triggers, such as infection, dietary supplements, exercise, and some medical supplements as well. The therapies for this TANGO2 disorders, wonderful work by Christina and her team has shown that vitamin B prevents metabolic crisis in these patients. There's a recent study in zebrafish also where it was shown that one of the compound calpeptine can also prevent some of the disease symptoms, but still there's no cure for TANGO2 deficiency disorder to reverse all of the symptoms. So that's why we need animal models to study TANGO2 disorder, especially because TANGO2 mice at the baseline, they exhibit normal function and lifespan. So mice is a common choice of model to study disease and develop therapies. When we started this work, there was really no animal model out there that can recapitulate most of the symptoms that are seen in these patients. So we decided to develop a zebrafish model for TANGO2 deficiency to study the disease. Before I go into zebrafish, one of the questions that many of the patient families have, why do we need so many different animal models to study one disease? And each of these models is unique in a way. Sarah talked about worms. I'm not going to talk about much, but you'll be hearing talks on worm, flies, fish, and mice today. When we decide to study any of these animal models, one of the first questions we ask is the disease-causing human gene present in the animal model. And we are lucky in the way that TANGO2 gene is present in all of this animal model, and each of this model presents certain advantages over the others. The next question that we ask is, the gene that we want to study, how similar it is to the human gene? Because higher is the similarity, higher is the chances that you're going to see the disease symptoms that we see in the human patients into these animal models. And a disease like TANGO2 deficiency disorder, which is multisystemic, we also want to see the different tissues in this animal model, how similar are they to the human patients? And finally, what kind of therapies can be developed? For example, worms, fly, and zebrafish, they're really good to test a large number of compounds. We can test thousands and hundreds of compounds in these animal models, and Sarah talked about some of that work their lab is doing. On the other hand, mice is very similar in terms of the cellular structure to the humans, but we are limited in the number of therapies we can test at a certain time. We can test only 1 or 2 therapies. So using a combination of this model, we can test a large number of therapies by these different animal models. So now I'm going to talk more about zebrafish, why we are working with zebrafish. Zebrafish are tiny organisms. These are vertebrates about 2 inches in size, and you can buy them for a few dollars at any pet store. The reason we do not study for that reason, the reason we study for, because 70% of the zebrafish genome is similar to human. So a lot of disease-causing genes that are present in human are also present in zebrafish. TANGO2 gene shows about 70 to 80% similarity to the human TANGO2 gene in zebrafish, and they can be bred easily. We can get hundreds of embryos just within a day of mating this zebrafish. So that way we can analyze a large number of embryos for any experiment, or if we want to test number of drugs, we can put one fish into each well and put one drug into each of this well to identify a handful of drugs. And more importantly, most of the organs that are present in the human system are also present in the zebrafish. Within a day of zebrafish development, they have heart, brain, muscle, all the equivalent of the human organs. And similarly, this is an example of the adult fish that has similar organs that are present in the human. So once we identify any small molecule or any therapy using this zebrafish model, I think this is, sorry. This is a repeat, but anytime we identify a small molecule using a zebrafish model, we can easily take those handful of compounds and test in the animal models like mice, and eventually, if they show any efficacy, take them to the clinical trial. So a few years back, when we decided to make a zebrafish model of TANGO2 deficiency disorder, what we did, we took zebrafish and created mutations in the zebrafish genome. So these are permanent deletions, and what do those mutations do? We do not see any TANGO2 protein in the mutant zebrafish. So they're very similar to the human patients, that these fish do not make any TANGO2 protein. And then we looked at this zebrafish under the microscope. So this is like really magnified view of the zebrafish. These zebrafish embryos are usually 2 to 3 millimeter in size when we see by the naked eye, but these are several hundredfold magnified. And one of the surprising thing for us was that we did not see any defect in this fish, which we thought was a big setback because we work with a lot of other neuromuscular diseases, and zebrafish models those disorders really well. But if you see, this is a control fish and this is a mutant fish. They look exactly similar. Then we next looked at the swimming pattern of this fish also. They were very similar in terms of swimming behavior. But then when we started to monitor this fish over a long period of time, what we found was this is the control fish. So zebrafish, like mice, can live up to 2 years. And the control fish, they survive. This is the 100% survival rate of the control or wild-type fish. But these are the mutant. And we see that some of the mutants start dying like right after 1 week. And eventually, most of the mutant die by 3 months of age. But a small percentage of this mutant do survive. And I'm going to come to that again later. So this is the quantification. This is both control, and these are mutant. In terms of body weight also. So we do not see a big difference, but by this age, we do see a small difference in the body weight. In the mutant, they have reduced body weight. So even though we did not see big difference in the swimming, the fish looks okay when we look under the microscope, we decided to look inside the different structure of zebrafish. And what I'm showing you here is the electron microscope image. So when the previous image I showed was 100 times magnified, this is thousands of times magnified image of the skeletal muscle in this picture. So we see this nice sarcomere organized in the wild-type fish. And when we look in this image, this structure is called mitochondria. And you have heard about it, and you're going to hear again about this structure in the mitochondria, which regulates the whole energy balance in most of the organs. So surprisingly, even though we did not see any swimming defect, the fish looks normal, we saw this mitochondria, they are broken down in the mutant fish. So this was very surprising. It means that there is this mitochondria that regulate the energy balance. This is the only defect we see. Could it be affecting some of the metabolites in the muscle or any other part of the body? So with that in mind, we decided to look at some of the lipidomics. But just before going there, some of you may have the question, if the mitochondria looks so bad, why the fish is swimming normally? So we did the quantification of the mitochondria, and what we saw was that it only a small percentage of mitochondria that are bad, not all the mitochondria are bad. So with that, because we have thousands of mitochondria in each cell, with a small percentage of mitochondria bad, they are still able to do normal function. So we next went and started to stretch this muscle. What happens if we stretch this muscle? Okay, in the normal form, they can swim longer, the muscle is okay. So we did an assay to cause stress in the zebrafish, where we induce exertion in this zebrafish. So what we are seeing here is a staining in the skeletal muscle. This is a normal fish with a normal staining. The muscle fibers look perfectly fine. And in this assay, what we wanted to do for the exertion, usually in the mice, when we want to exert the mice, we put them on treadmill for a few hours. And then they get exhausted, and we can start to see the changes. But one of the challenge for us was, what do we do with the zebrafish? How do we make them exercise? So then we came up with this idea, if we increase the viscosity of the water so that they have to work harder to swim, something like instead of swimming in the water, what if we make the solution like honey, where they have to work harder? So we added a polymer called methyl cellulose, and because of that, the water became really viscous and let the fish swim for an hour or so. So what happens is it's similar to us. When we work out after a long time on the treadmill, our muscle gets sore. So this is a control fish. You can see compared to the without exercised fish, this fish look little, muscle structure looks a little abnormal. But the most surprising result was from the TANGO2 mutant. We have 2 different TANGO2 mutant zebrafish, and both of them, we started to see this tear in the muscle. Muscle looks like really stretched and really tired. And most of the fish that were exercised, normal fish, they came back to the normal swimming after a few hours, but this fish took really long time to recover back. And on the top is, most of the fish look like this, but there were some fish where the whole muscle broke down, and this fish never recovered. They just collapsed and died. And this is the quantification. So this is without any methyl cellulose treatment. In the gray is the mutant, and this is the wild type. They both swim to the same extent. But when we make them swim in the methyl cellulose water, the mutants just crash. So this kind of tells us that, okay, in the basal state, we do have some mitochondrial defect, we do have some energy deficient, but when we stress these TANGO2 deficient zebrafish, that's when the muscle cannot handle after a certain point. So to quantify the changes that are happening in the skeletal muscle at that point, we performed lipidomics, where we took the whole TANGO2 mutant and analyzed all the potential lipid species that are present in the body at that time, and try to see what are the specific lipids that are upregulated or downregulated and could be contributing to this phenotype. And initially, we did this experiment in the absence of any trigger. So this is a baseline fish, and we saw that in the green, you can see these are all the lipids that show a big difference in the mutant zebrafish. So even at baseline, when everything is fine, fish have no defect, they're swimming happily, there are still inherent changes that are happening in the body of this fish. And then we looked at the specific, we broke down this total lipid into specific lipid subtypes. We found the phospholipids, phosphatidylcholine, and transglyceride and phosphatidylethamine. So there are three lipid species that were highly downregulated in the mutant. So, and Sarah talked about some of these. These are linked to the acetyl-CoA pathway, but then these phospholipids are also critical for a number of other processes in the cell. Most importantly, they are required for the formation of cellular membranes. So if these phospholipids are not there, cells can function in a certain way, but when there's a stress present on the cells are going to break down and result in different kind of deficiencies. So after exercise, as we all know, TANGO2 patients have a number of different phenotypes. We wanted to see what are, can we use the zebrafish model to identify different clinical phenotypes? What are the clinical phenotypes that are present at the baseline level? And what are the clinical phenotypes that can appear when we stress the zebrafish by different stressors? So another clinical symptoms associated with this patient is a development of seizures during their lifetime. So at this point, we collaborated with Dr. N. Paduri, who is an epilepsy specialist at Boston Children's Hospital, and her lab studies zebrafish model of epilepsy. And what they have done is they have developed this assay. When they treat the zebrafish with this compound called 5 millimolar PTZ, zebrafish model that have epilepsy gene, they show a big difference than the control fish in terms of their spontaneous activity that they can be quantified. But when we treated TANGO2 fish with PTZ, we did not see any difference. So the blue dots here are the mutant, and the green and orange dot here are the control fish. And this can be seen here. where we broke down in a number of assays to see if this fish have any susceptibility to develop seizures. But looks like at the earlier stages, this fish they do not develop any seizure when we stress them with the chemical compounds that can otherwise cause seizures in the epilepsy models. So this is another phenotype I want to show you. As I mentioned, some of these fish survive and live up to adulthood. So I want to show the first video first. Oh. Okay. So in the first video, you see two fish swimming. On the top is a normal wild-type fish, and at the bottom is a TANGO2 mutant. Now these are the adult fish, and you can see that the wild-type fish is going in the straight lines from one end to the other end of the cage, but the mutant fish is just staying at the bottom. And this is now another TANGO2 mutant. So this was really surprising that when they're younger, when we expose them to chemicals, we do not see any seizure. But as they get older, they start to develop this spontaneous seizure-like dysfunction. And as they collapse, most of these fish that develop the seizure, they never recover. They eventually die within few hours. So now we are at that point that we are trying to understand how, at the base level, these brain abnormalities are appearing in the zebrafish by analyzing them at different time points. This is just a quantification of the videos I showed you. On the left, we have a normal fish which just goes in straight lines, swims, but this is a mutant. When they have the seizure activity, they just go in the circle and eventually collapse. We also looked at the heart function in the zebrafish using a unbiased AI-based approach where we use this high intensity cameras that can record the heart function and quantify the heart function, and we did not see any defect in the heart function at the base level. So just to remind you that we have a few fish that survive to the adulthood, and what we are doing right now is using that to develop therapies in zebrafish. So most of the TANGO2 fish are sick. They die by 3 months of age. But I mentioned previously, we have 3% of the fish that survive, and they survive up to 2 years of age like normal fish. We have compared the DNA for these two fish, and now we have identified a number of variants which are specifically present in the escaper fish. And our next goal is to screen those variants one by putting them back in the sick fish and see if we can make TANGO2 fish better using this approach. So I think I'm running short on time, so I'm going to go fast to finish my talk. The second approach that we are taking is using small molecule screening in zebrafish. So this fish have defect in the mitochondrial function. We have identified a library of small molecules that specifically target mitochondria, and using our previously developed assay where we can add one drug into each of this compound and quantify the swimming behavior, we hope to identify small molecules that are making the TANGO2 fish better. And this is an example of a screening that we have just finished recently for another rare neuromuscular disorder. Each of this dot is a zebrafish. So when we expose this fish to different drugs, this is a control fish, we can see an increase in the swimming capability of this fish. So we are hoping to identify some compounds in TANGO2 zebrafish model using this approach. Just to summarize what I presented today, a wide range of animal models are needed to understand the disease and develop therapies for TANGO2 deficiency. Zebrafish model of TANGO2 deficiency closely recapitulates the disease at several levels. We see several changes happening at the baseline, as well as when we expose them to certain stress factors. And a number of therapeutic developmental effects are in pipeline for TANGO2 deficiency disorder. So with that, I would like to thank, I think my last slide didn't show up, all my lab members and TANGO2 Foundation for supporting this work. Thank you.